Hi everyone. So today we are going to talk about animal reproduction. There was a question last year in mains paper 1 on animal reproduction, specifically reproduction in sponges. So how reproduction in sponges happens? Is it just sexual or asexual? And what are the various ways in which sponges can reproduce? So we are going to discuss that today. So let's discuss a little bit about what are animals first and foremost, and very quickly we'll jump onto sponges and reproduction strategies in sponges, and how are we supposed to respond to this question. So it's a 15 marks question that was asked last year, and a 15 marks question should ideally take you 9 to 11 minutes or maybe 11 to 13 minutes max for answering. You will not get time more than that on an average in your mains paper 1, and it should be approximately 250 to 300 words, not less than that and not more than that. That's the word limit for it. So that means the thing should be very crystal clear in our head, and we just have to pour it down on the paper. We don't have much time to think. Okay, let me show you how does 250 words fit into the paper if I make a flow chart of it, and if I talk about reproduction in sponges. Let's first see that, and then we will talk about the details of this. So this is reproduction in sponges. I have made a flow diagram of this, and this flow diagram is containing approximately 250 to 300 words. There can be certain changes in this flow diagram depending on the kind of answer sheet that we are getting. We can break it into two parts, of course. I am pretty sure we don't have such a wide answer sheet. So obviously we will have to break it into two, right? So here, since I have the freedom of utilizing the vast space of this board, so I will be doing that. Okay. So talking about sponges, they are one of the simplest animals that we find around us. Not around us, in fact, most of them are sea dwelling. There are very few freshwater sponges known to mankind. Approximately 8,000 to 10,000 species are there, not more than that. As of now, the numbers will increase surely, and some might become extinct, the new ones might be getting discovered. So all those things are there, of course, but right now that's the situation. Okay, so talking about reproduction strategies in case of sponges, they have complex reproductive strategies that enable their survival through asexual and sexual methods of reproduction, ensuring their continuity, their success. Okay, but I find with many people a difficulty in understanding what is asexual reproduction. So first and foremost, we are going to talk about asexual reproduction a bit in detail, and so I have shown you how the answer will look like. And okay, let me show you a few more answers. So this is one way of putting up the answer. This is approximately 250 to 300 words. Another way that you can put up your answer is this. This might fit into your sheet scheme. And this is approximately 200 words. So maybe 2 to 3 lines above this, 2 to 3 lines below this will make your answer complete. So 250 words is not that much of a stuff as it sounds. Okay. Fine. So these are the reproduction strategies in sponges. Of course, this column is slightly mistaken. This has to be shifted here. This is a kind of sexual reproduction strategy in sponges, not asexual reproductive strategy. Okay, talking about asexual reproduction. One of the common problem that I find with many of the learners of zoology also, that they say asexual reproduction involves one parent and sexual reproduction involves two parents. Well, in many cases, sexual reproduction does happen with a single parent organism. Let's take a plant which is bisexual. That means it has got both a complete flower. That means the male as well as the female reproductive parts in the same plant. it can undergo self-pollination, and thus it does reproduce sexually with a single within a single organism. Sexual reproduction is happening. So the definition of asexual reproduction, that it happens in one parent, stands void here. So how should I say how should I define asexual reproduction or understand it? Asexual reproduction does not involve fertilization or fusion of gametes. That thing is not going to happen. Gamete formation might happen. The gametes might give rise to an animal parthenogenetically. Like in case of honeybees, male drones are produced asexually from the unfertilized egg cells of the queen. So gametes are there. In many cases, gametes are, in fact, in most of the cases, gametes are not even required in asexual reproduction. You don't require germ cells also. Right. So what are germ cells? What are gametes? I hope we are pretty much familiar with these terms, with this basics. So I'm not going to dwell on that, and I'm going to dwell on the answer now. Okay. One of the common things which is used in case of animals is regeneration. And this regeneration is something that we find in vertebrates also. But in vertebrates, regeneration, like in case of lizard, if you chop off its tail, it gives the lizard an ability to regrow its tail, a wall lizard. It's a very common example of regeneration. Or a starfish losing one of its arm and then having it back after a few days or some time. So that is another example of regeneration. But in lower non-chordates, in fact, I prefer to use the term non-chordates rather than invertebrates, because some chordates does not have a vertebral column. They are chordates and they never ever in their whole existence have the vertebral column. So technically they are invertebrates, right? So there is a confusion of terminologies also between non-chordates and chordates. Is a more appropriate terminology compared to vertebrates and invertebrates. Okay. So sponges are non-chordates. That means they do not have notochord at any point of their development at all. Okay. So asexual reproduction in case of sponges. If I take a sponge and if I grind it like I am grinding ginger, and after that I leave it, it can propagate again in sea and form new sponges. So that's the power of regeneration in case of sponges. So they do have a great power of regeneration. Of course, the lost parts can reappear back. That is very much there in case of sponges. But of course, it is a good way of reproduction also. But regeneration is not a very wonderful way of asexual reproduction for most of the animals. This gives them the capacity to survive in case of mechanical desiccation. But this is not their preferred mode of reproducing asexually. In fact, budding, as we find in case of hydra very commonly, is a better and a preferred way of asexual reproduction in sponges also. Okay. Then we might also see branching. So budding, new buds are formed like in case of hydra, right? Although hydra is not a sponge, it's a coelenterate. Or in fact, currently we don't use the word coelenterate. The scientific community prefers using the word cnidarians instead of coelenterata. Although both can be replaced to a lesser or a greater extent. So budding, the most common example is hydra. If you talk about fungus, you see example of yeast, a unicellular fungi which produces buds. So that is another thing. Okay. So branching. Sponges, regeneration happens, and for regeneration, for budding, for branching, one kind of cells are very important, and they are called as archaeocytes. And you might also call them as amoebocytes. And in biology, you will often find the two suffix being used pretty often, sites and blast, for the name of cells. The blast word is generally meant for immature cells. The site word is generally used as a suffix for mature cells. So both the words archaeocytes and amoebocytes are more or less replaceable. They mean one or the same thing. These are like totipotent stem cells in case of your sponges. And these cells can transform into any kind of cell of a sponge. Maybe a pinacocyte, maybe a quanocyte, maybe a egg cell, right? So it can transform into literally anything. Sponges, remember, they have cellular level of body organization. They do not have tissue level of body organization. So that is something to be remembered. Okay, moving down to the next thing, fission and fragmentation. So fission, it's not binary fission like in case of amoeba or like in case of bacteria. So here fissions or fissures appear on the body of the sponge over a period of time, and these fissions or fissures or these cracks actually allow a part of the sponge to fall off, and once it falls off, if it has got archaeocytes or amoebocytes, it is completely capable of giving rise to a new sponge. Very simple. All right, moving to reduction bodies. What are reduction bodies? Reduction bodies are basically, see, harsh environment is there, the sponge is not able to reproduce, not able to even survive, forget about reproduction. So when it is not able to survive, only a part of it is there, and that is called as a reduction body. So reduction body is basically nothing, you can say a group of archaeocytes which are totipotent amoebocytes. Instead of showing them as round, I should show them in an amoeboid shape. Means in a shape of like an amoeba, like the way we draw an amoeba cell. That's why they are called as amoebocytes. That means they don't have a rounded shape. So we will have a bunch of archaeocytes or amoebocytes, and they are surrounded by a bunch of pinacocytes. And that's it. That's a reduction body. So this is going to sustain the harsh conditions. The rest of the sponge is dead, and when the time is appropriate, this reduction body can give rise to a new sponge. Okay, moving forward. So if we are in a live class, the benefit is that we can quickly ask questions. I know many questions are heading in my mind also as a student right now, right? But we can engage more in a live class. In a recorded lectures, that is bit of trouble. The live questions, picking up live questions. Okay. All right. So let's talk about gemmules. Gemmules are also called as survival pods. So these are generally found in mostly freshwater sponges. So there are very few freshwater sponges. So their gemmule formation happens instead of reduction bodies, which are found mostly in marine sponges. So marine means, of course, sea dwelling, you know that. Okay, so survival pods. What is difference between a reduction body and a gemmule? So in case of a gemmule, again the gemmule formation happens once the sponge is desiccated, it is dying, it cannot grow, right? Only a small part is left. But this is a more specialized structure, gemmule. Here, of course, in the center you will have the archaeocytes, the totipotent stem cells as you would like to call them. I am showing them round instead of amoeboid in shape. Pardon me for that. And then, of course, we will have a layer, a tough layer, a chitinous layer, instead of pinacocytes, which were, so here the archaeocytes were lined by pinacocytes. This was pinacocytes in case of reduction bodies. In case of gemmules, these are not pinacocytes, this is a chitinous cover, a tough cover, and then you will have lots and lots of spicules. So this is the inner tough cover. You will have spicules. You will have a less tough outer cover with an opening. So the opening is actually let's draw the opening here. So this opening channel pierces through the tough as well as the inner and the outer cover. So the inner cover I am showing with the red color, which is made up of chitin. The outer cover is made by yellow color, and we can see this opening. This is called as micropyle. This word you will see very often in botany also. Micropyle seems to be the word over there also. So this micropyle is covered with this loose layer, which is the yellow one, the outer cover. Let me label it. Just writing OC for outer cover. We have this inner cover. Now, there are spicules, which I have shown with the blue color. That's a gemmule, a very specialized structure found mostly in freshwater sponges, including spongilla. Okay. And so these are spicules. So that is gemmule, and it can survive. Okay. So more or less we are done with asexual reproduction in our so-called plants, sorry, not plants, sponges. And let's talk about sexual reproduction. So if I am talking about sexual reproduction obviously means you will have two gametes. In this case, the gametes are not isogamous, they are anisogamous. In fact, it is appropriate to say the type of gametes is like, in this case, we will have oogamy. So we have a ova or the egg cell which is produced in the mesohyle or the mesoglia of the sponges. How? From the archaeocyte. And that stays there. So sexual reproduction involves fertilization. Fertilization in almost all cases is internal. It is rarely external. So fertilization is internal. All sponges, okay, few words we can add here. First and foremost, all sponges are hermaphrodites. We don't have separate male and female sponges. Every sponge is capable of producing both kind of gametes. Okay. So ova is produced from the archaeocytes, which remain in the mesoglia. Sperms are produced from the choanocytes or archaeocytes, as the case may be. Now what happens? The fertilization is mostly internal, as I told you, rarely external. Now the sperms which are produced by one sponge will be released into its spongiocele. Through the spongiocele, through the osculum, it will be pumped out along with the water. And once it is pumped out, it will go to a nearby sponge. Does self-fertilization happen in sponges? I don't find that in literature much, right, anywhere. So I'm pretty doubtful about that. So nowhere you will see self-fertilization being mentioned in case of sponges. So we can say it is cross-fertilization. Okay. So the sponge is taken up by the ostia, the dermal ostia, because we know the inlet root is the dermal ostia in for all the sponges. It is not the osculum. Osculum is the outlet root. So osculum ensures that the sperms go out. The ostia ensures that the sperm from the sponge 1 goes into the sponge 2. And of course, it will enter the spongiocele ultimately. And once it is there in the spongiocele, it will be caught by choanocytes of the choanodome, which is lining the spongiocele. Now, once it is caught by the choanocytes are going to consume this, eat this sperm, ensuring that its genetic material, its nucleus, its important components are safe. So it doesn't digest it. It just eats it, right? That means phagocytize it, but does not digest it. So the phagocytic vacuole does not means continues to save the sperm inside the choanocyte. This choanocyte will migrate to the mesoglia. And once it migrates to the mesoglia, the choanocyte is releasing or is getting swallowed by the ova over there. And once it is swallowed by the ova, the genetic material of the ova and the sperm fuses, and we have the fertilization achieved. So the fertilization is internal. Sperms enter sponge body, they are trapped by choanocyte, delivered to ova. Fertilization is internal. Now, development is indirect. Since there are larval stages of development, we say the development is indirect. It's not direct. So you can use these terms in a para or in 2 to 3 lines while you are giving an intro, right, or maybe beneath it. So the development in sexual stages is always indirect, it is never direct. Now, what is holoblastic, radial cleavage, blastula, gastrula, all this? So we have learned a little bit about animal embryology. So once fertilization is achieved, we know we have a zygote. This zygote undergoes cleavage, right, which is slightly different or not slightly, in fact, I can say is different from mitosis, right? So, of course, slightly different, you can say. Why slightly different? Because here the number of daughter cells that are produced are always having less protoplasmic content compared to the parent cell, right? That doesn't mean the genetic material is less, but the other cellular components are surely less. Okay, so the zygote will undergo cleavage, it will form a morula stage, a blastula stage, and this blastula generally undergoes the next stage, that is gastrula stage, during the embryological development of an animal. And during the gastrula stage, we see developments of germ layers. In case of sponges, there are no tissues. It is just a bunch of cells. So, although there are specialized cells, pinacodome made up of pinacocytes, choanocytes, archeocytes, sclerocytes, you name it. So, there are 20 to 30 different kinds of cells that one can talk about, at least 10, which are there mentioned in your literature, right? So, but all those cells are derived more or less from amoebocytes or archeocytes. There is no germ layer formation as such during the so-called gastrulation event in case of sponges. Therefore, any literature mentioning that they are diploblastic, that is incorrect. Because unless and until you have the germ layer formed during gastrulation or post-gastrulation, you cannot talk of diploblastic or triploblastic. You cannot say sponges are acoelomates. That concept of coelom or pseudocoelom or no coelom does not stand valid for sponges. Why? Because they do not have germ layer formation during gastrulation. So that is to be understood. What is holoblastic cleavage? That means the zygote divides completely, right, into two. Radial cleavage. This is one axis, the second axis, the third axis. So along the radii, there is radial cleavage and spiral cleavage. You would have learned about that during embryology. So the cleavage is radial. Okay. Now, we cannot talk about sexual reproduction based on the classes in sponges. It will be more appropriate to talk about sexual reproduction in case of sponges based on the type of canal system that they have. In some literatures, people have written vascular system. Sorry, water vascular system is present in echinoderms, not in sponges. In case of sponges, it is water canal system. So don't get confused, please. Okay, moving forward. So we know we have three kind of canal system, and what are they? Basically three kind we mentioned broadly: ascenoid, cycloid, leuconoid. Right. So we are talking about cycloid canal system, which is slightly more complex compared to the simpler ascenoid canal system. Right. So sponges that are having cycloid canal system over there, during their blastula stage, they will have the blastula which is said to be, which is called as a stomoblastula. The word stomo means mouth. So during the blastula stage, so here the first stages of larval development are happening in the mesoglia of the sponge. They are not happening outside the sponge body, right? So the larva migrates from the mesoglia and ultimately it is ejected out by the sponge body, and the later stages of larval development, generally gastrula onwards, are happening outside the sponge body. So that is one thing. Okay. So the stomoblastula is a kind of stage, a blastula stage, where the blastula, I think we all know blastula means you will have the blastocele, you will have the trophoblast cells, right? You will have the inner cell mass, that is the blastula stage. So here the blastula stage also seems to have a mouth, and it floats in the mesoglia, and it feeds through its mouth on some nerve cells which are meant for providing nourishment to this larva. So that is why, since it is having that mouth like thing, so for mouth we are using the word stoma or stomo in this case. So this is stomoblastula, and what happens, then this stomoblastula inverts itself. So its mouth is there, it is picking up stuff, and then it inverts itself completely. It undergoes a transformation. It loses its mouth, and now it is amphiblastula stage. And during the amphiblastula stage, once it inverts itself through its mouth, that means the outside portion comes inward and the inside portion comes outward. The next thing is gastrulation. And now, during the gastrulation stage, the larvae has flagella on the outside. Now it has moved out of the sponge body. And now this larva can move, it is mobile, it will settle itself on a suitable substratum for its growth. And then, after the gastrula settles itself, it goes into the next stage, which we use for all the sponges, which is called as Olympus stage. That means a single cylinder of a sponge having its water vascular system with a osculum, with a spongiocele. So one cylinder comes up, and that is the Olympus stage. So that is the way the cycnoid sponges reproduce. Then we can talk about other sponges also after cycnoid. Spongilla has a special case. We'll talk about that. And but before spongilla, one thing is missing in this picture, which I would like to put up. Okay, I have deleted one of my slides, so that was not there. Okay. So what about ascanoid and leuconoid? How do they reproduce? In case of ascanoid and leuconoid, that is not mentioned here. So since this is 200 words, you can have another 220 words or 230 words by adding information about ascanoid and leuconoid, which I have not covered in this table. Okay. So talking about reproduction strategies in ascanoid and leuconoid canal system sponges. The first blastula stage happens. After the blastula, there is gastrula stage, right? But here, the blastula, the gastrula stage is not hollow. And in fact, the blastula stage is not hollow. It is called as coeloblastula. Right. And this coeloblastula converts itself into the gastrula stage. See, names are there. And these names are to be remembered. So basically, the UPSC aspirant is being tested on its memory more. Right. So even I tend to forget the names unless and until they are there in front of my eyes. And since the name is missing, so the blastula stage is there, coeloblastula, and this undergoes gastrulation. And in gastrulation, there is no germ layer formation, remember? And it is this gastrula which will be mobile. Right. So remember, there is one term which is very oftenly used, sessile, which means unable to move. So most of the coelentrates, right, that means especially their polyp stage, and all sponges are sessile as an adult. But during the larval stages, the gastrula stage seems to have the flagella outside. And it can surely reproduce. Right. So I hope this is clear. Let's get back to our diagram or slide, whatever it is. Okay. So, okay. There was one that I have missed out. That was containing sexual reproduction here, which I wanted to list here. That was mentioning about ascanoid and leuconoid. And that was the information I wanted to share with you. In spongilla, the case is slightly unique. The gastrula actually appears like a tent. It is mobile, it settles on a substratum, but appears more or less like a tent. Right. And it has got a means sections called as hypophore and spongophore. So hypophore seems to be its the spongophore develops into the osculum. The hypophore is its lower portion, the foot portion of this larva, and then it goes into the olantha stage and forms an adult. So that's a very brief overview, but I have shown you how to write the answer. If UPSC appreciates your answers in the form of basically flowcharts, that's wonderful. But my point over here is one should not be forgetful. One has to make, first and foremost, understanding is important. Once the understanding creeps in, the next stage is summarizing or noting it, writing it down, the important points, right? And once you have written it, the next step is practice. So you will have to practice this, how to write it in different possible formats, highlighting at least the key words, maybe just by underlining them, right? So that is very important. So your diagram might not be having these colored fillings, which is absolutely okay. So we are not going to mimic a book or a PPT over there. That is not the intent. They just want to know whether you remember the terms, you understand the concepts, and whether you can deliver it in a tight deadline with a lot of pressure on your head. So that's the crux of asking such questions. Mostly the contentious questions like the phylogeny of an organism, especially in case of sponges and all, will be avoided. Those questions which are straightforward, have a straightforward answers, will be generally asked, right? Because phylogeny is more or less debatable. So we can discuss about phylogeny. They might ask you about structure of sponges. There is no contentious issue about that. 3 layers, mesoglia, mesohyle, whatever name you would like to use, and then the middle layer is non-cellular, primarily a matrix of spongin fibers, gel-like substance containing spicules, etc., and a lot of cells, archaeocytes, amoebocytes, etc., right? And of course, we have the pinacoderm and the choanoderm. So generally, the questions are asked which are non-contentious even from a subject point of view, right? So they just want to check you on your ability to retain what you have learned and perform well under pressure. That's the moral of the story. So I hope the message is clear. And this was one question that was asked last year, mains paper 1 for 15 marks, and we have discussed it. And we can discuss a lot of other things also, but maybe some other time. Thanks a lot.